Hello everyone, and welcome. This presentation is brought to you by AZCare. Together, we'll explore the world of scientific publications, breaking down complex research into clear and meaningful insights. Welcome to Decoding Science. Today, AZ will guide us through a fascinating study on how specific brain changes affect memory, which is highly relevant to understanding conditions like dyslexia. Imagine trying to remember something important, but your brain just will not hold on to it. Today we are exploring a study that helps us understand why. AZ, what is this paper about?
This paper is about how specific brain changes, called microgyria, affect spatial working memory in rats. It helps us understand persistent memory issues, especially those linked to human conditions like dyslexia.
What key information will we learn from this discussion?
We will explain what dyslexia is, discuss brain anomalies like microgyria, describe how these were studied in rats, and share the important findings and their implications for future research.
AZ, to start, can you explain what dyslexia is?
Dyslexia is a reading disability that affects about 5 to 10% of children. It is often linked to difficulties in processing rapidly changing sounds.
The paper mentions ectopias and microgyria. What exactly are these brain anomalies?
These are collections of brain cells that did not move to their correct locations during development. Such anomalies, specifically ectopias and microgyria, have been found in the brains of individuals with dyslexia.
How do researchers use animal models to study these brain disruptions?
Researchers create similar changes in rodents, which are called induced microgyria. This allows them to study the behavioral effects of these brain disruptions and gain insight into human conditions.
What other cognitive challenges are often seen in people with dyslexia, beyond reading difficulties?
Dyslexia is also characterized by deficits in both short-term memory and working memory, which includes challenges with verbal and visual spatial information.
What did prior studies using rat models with microgyria reveal about their cognitive abilities?
Previous studies showed that microgyria in rats leads to specific problems with rapid acoustic processing, mirroring deficits seen in human dyslexia.
Given these connections, what was this study's primary research question?
This study investigated whether rats with induced microgyria also exhibit persistent spatial working memory deficits, aiming to reflect aspects of human dyslexia. Before diving into the paper, let me introduce AZCare. Americans spend nearly 1 billion hours on hold each year. AZCare's HIPAA-compliant AI agent, AZ, saves time by booking appointments, resolving billing issues, and more.
Now that we have covered the background, let us explore the precise methods researchers use to study working memory in rats with microgyria. Back to the paper. AZ. How was spatial working memory specifically tested in this study?
The researchers tested spatial working memory using a delayed match-to-sample radial water maze. 8 microgyric, or MG, rats and 10 sham-operated rats participated in the experiment.
Please describe the radial water maze task in more detail for our listeners.
Certainly. In a sample trial, rats learned the daily location of a submerged platform in one of 8 arms. Then, after a 1-hour delay, they were tested. The goal arm location changed every day, so rats had to use their working memory for each new task.
Figure 1 shows a diagram of the maze. What exactly does it illustrate about the task setup?
Figure 1 illustrates the maze setup with changing start points and goal locations for the sample and test trials. The rats' performance was measured by the number of incorrect arm entries, or errors, over 60 testing sessions.
With the methodology clear, let us now examine the study's key findings on how microgyria impacted the rats' memory performance. AZ, what did the brain examinations reveal about the MG rats?
Histological examinations confirmed that all 8 MG rats had microgyric malformations, which were visible as small indentations and abnormally layered cortex, as shown in figures 2A and 2B.
How did the MG rats perform on the maze task compared to the sham rats?
As shown in figure 3, MG rats made significantly more errors than sham rats when trying to find the platform during the initial testing phases.
Did this memory impairment eventually resolve, or was it a lasting issue?
This memory deficit was surprisingly persistent. It lasted across all 12 weeks, or 60 days, of continuous testing, showing a sustained impairment despite both groups learning over time.
Did the MG rats try to solve the maze using a different strategy than the sham rats?
Interestingly, Figure 4, which displays pie charts, shows that their first error patterns were similar to those of the sham rats. For example, they frequently entered the previous day's goal arm or an arm adjacent to the current goal.
What does this suggest about the nature of their memory deficit?
This implies that MG rats were indeed using the same spatial strategy as the sham rats. However, their memory representation of the correct goal location was simply weaker, leading to more errors.
Was swimming speed or any motor impairment a factor in the MG rats' increased errors?
No, we found no differences in swimming speed between the groups. This indicates that the increased errors for MG rats were due to a cognitive deficit, not a motor impairment or a general delay in movement.
Finally, let us connect these important findings to our understanding of dyslexia and explore the exciting directions for future research. See, what was the most significant finding regarding the MG rats in this study?
The key finding is that rats with induced bilateral cortical microgyria exhibited persistent deficits in their spatial working memory. They consistently struggled to remember spatial locations over time.
Are these findings unique, or do other studies support this conclusion?
These findings are consistent with prior research on ectopic mice models, which also demonstrated similar memory anomalies and problems with spatial tasks.
What do researchers believe might be the underlying cause of these memory issues?
The underlying cause is thought to involve a widespread reorganization of neural circuitry. This reorganization occurs because of the early disruption of cortical neuronal migration during brain development.
How do these findings in rats relate to our understanding of human dyslexia?
Working memory deficits are a critical characteristic of human dyslexia. So this rodent model provides valuable insight into the neurobiological basis of the condition and its developmental etiology.
Are these working memory problems in MG rats linked to any other issues previously observed in them?
It is highly possible that these working memory deficits are connected to the rapid auditory processing problems that we observed previously in similarly treated MG rats, suggesting a complex interplay of issues.
What are the exciting next steps and future directions for this research?
Future research will explore connections between dyslexia risk genes, like DYX1C1, and working memory deficits. We will also investigate potential changes in the hippocampal structure in MG rats to understand the full impact of these brain anomalies. Thank you all so much for your time and attention today. I truly appreciate your interest and engagement throughout the presentation. I hope you found the discussion insightful and that it sparked some useful ideas or perspectives. I'd be happy to take any questions you might have or hear your thoughts and feedback.